Now that we've discussed some of the small-scale chemistry that you need to know about, we get to start talking about some of the more biologically relevant aspects of chemistry. So we're going to start talking about those macromolecules, those building blocks of living things. And these are building blocks of living things because they're molecules that we can get energy from and store energy in. In addition to that, they're going to play structural roles. They're going to play roles for communication and identification. So lots of jobs within the body because, well, our body has to do lots of different things in order to be alive. At their core, all of them are going to have carbon, hydrogen, and oxygen somewhere in their structure. And in particular, the carbon and hydrogen will form a backbone or a skeleton that everything else will attach to. Because they have some of these common characteristics, we're going to see that there's some common terminology to go along with that. We're going to see that these are referred to as polymers, which means they're chains with many pieces in them very often. And each individual part of that chain is going to be referred to as a monomer. So I think of a lot of these as being formed almost like a necklace of pieces. And that, if there's a necklace of pearls, then every pearl would be a monomer and the whole necklace would be a polymer. We're also going to see an example where these molecules are isomers. They have the same parts, but they're in different arrangements. And this is very much like if you take a look at your hands. Your right hand and your left hand have the same parts, but they're arranged differently, which makes them mirror images. And that's actually really important in chemistry because if the shape doesn't fit exactly into another molecule in a chemical reaction, the reaction won't take place. And so even something as simple as being a mirror image of the right molecule can prevent two compounds from interacting. So shape is a really big deal. The first macromolecules we're going to talk about are the most basic ones, the carbohydrates. You guys are probably familiar with carbohydrates in your lives. We eat them because they give us energy. We think of them as fuel, but they also play some other roles. We can actually have some storage forms of them, like starches that we consume or complex carbohydrates that we talk about. And in some cases, they actually provide structures for cells or organisms. There's a general formula when we talk about carbohydrates that tells you that everything's kind of consistent. There are occasionally carbohydrates that don't follow these kind of rules, but you'll see something very similar to this in all cases of carbohydrates. That for every carbon the carbohydrate is made with, there should be roughly 2 hydrogens and 1 oxygen. In addition, the carbohydrates are typically hydrophilic, meaning that they're water-loving. That philic ending, if you might be familiar with the phobic ending, water-fearing, but these ones are hydrophilic, water-loving, kind of like Philadelphia is the city of brotherly love. The simplest carbohydrates are called monosaccharides, and they're considered simple because they're one piece of sugar. They're chains or rings of sugar, and they're good for really fast energy because they're easy for us to break down and harvest the energy out of those bonds. Some good examples of those are sugars like pentoses. See that they have 5 carbons, so then they have 10 hydrogens and 5 oxygens. That follows that general formula we discussed. 2 good examples of pentoses are ribose and deoxyribose. And these ones are actually kind of structural carbohydrates. We don't use them for energy. We use them to build DNA and RNA. And so DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. It's a reference to the sugars that we find in those particular structures. The other common sugars in this group are called the hexoses. These have the formula C6H12O6. Again, you can see that they're following that general formula that we've discussed. And the examples we talk about are glucose, fructose, and galactose. Glucose is the sugar that's most commonly used by the body. But remember I talked about some molecules being mirror images of each other? If you look here at glucose and galactose on this line, you'll see that the OH and the H in these structures are flipped around. That's basically the equivalent of a right hand and a left hand and how your thumb is on the opposite side of the hand. So your brain prefers the galactose shape over the glucose shape. And all the galactose shape really is this very slight rearrangement of the glucose molecule. Fructose, on the other hand, is more significantly different because if you compare the sections up here, you'll see that these two groups attached to the carbons at the core are actually flip-flopped. And so those molecules are more unique compared to glucose than the galactose is. From those monosaccharides, you can then build disaccharides, two-piece molecules. Sucrose, which we commonly know as table sugar, lactose, which we commonly know as milk sugar, and maltose, which you might call beer sugar. That's the malt that we get from barley. It's less something we build than it is something we break down. So when you have really complex carbohydrates like these polysaccharides, you'll break them down first into disaccharides like maltose, and then finally into simple carbohydrates like glucose and fructose. And so that's a big part of like the beer making process, is malting the grains, is helping those starches break down from very long chains of glucose into little two-piece chains that are more functional. Polysaccharides are a little bit more diverse in terms of what they can do. We've got plant versions and animal versions that are relatively unique. And so amylose, which you probably know as starch, is the plant version of our storage sugars. Our own version as animals is glycogen, and we see an example of it here. There's probably about 60 molecules of glucose chained together in this glycogen, and that would be stored in your liver, so it's your storage version. It's like animal starch, if you will. Cellulose is a structural carbohydrate. We see it in the cell walls of plants, and in our diets, we refer to it as fiber because it's the part that we can't really digest and break down in our diet. And then the animal analog to that is chitin. It's found in the exoskeletons of insects and also in the cells of fungi. And fungi are kind of interesting in a lot of ways, but a lot of people tend to kind of clump them in with plants, that mushrooms are plants. When really, fungi are not plants at all. They eat things to get their energy. They don't make their own food. And here's a molecular example of why they fit better with the animals than they do with the plants, in that their chemistry is more similar to animals than it is to plants. In both these cases, cellulose and chitin, this is an example of creating an external skeleton for a structure rather than the internal skeleton that's more common in animals, for example. Now we're going to move on and talk about the lipids just a little bit. Lipids are different than the carbohydrates in almost every possible way. They do not have a consistent formula. They are hydrophobic, and they have a lot of different functions related to the very different structures that make them up. There's three big types of lipids: the fats and oils, which we're most familiar with, the steroids, which act as chemical messengers within the body, and phospholipids, which are part of our cell membranes. I'm going to talk about the phospholipids first because they're going to give us our best grasp on the monomers or pieces that these are typically built from. And phospholipids do not create huge chains, but they create these smaller chains where you have a glycerol molecule that forms the backbone of the structure, kind of. And glycerol's formula you'll see there is C3H8O3. It's sugar-like, but doesn't follow that 1:2:1 ratio quite exactly. And your body would treat it as a sugar if it wasn't participating in this molecule. And then there's these long fatty acid chains. They're big chains of carbon and hydrogen connected to a group that's called a carboxyl group. That's a carbon, two oxygens, and a hydrogen. We call that carboxyl. And this is an important group to be aware of because it makes things acidic. And so this is why this is a fatty acid, because it has that carboxyl group on it that gives it the characteristic of acidity. And lastly, we have a phosphate group. And it's kind of the oddball of this whole thing in that the fatty acids are both going to be the hydrophobic portion of this molecule. But then the phosphate group is actually hydrophilic. And so here you have a molecule that has both parts that like water and parts that don't like water simultaneously, which means that they will automatically want to align with one another. So let's think about this. Your environment surrounding your cells is mostly water, and the environment that is inside your cells is mostly water. So the part of this molecule that is hydrophilic is going to want to face the water. That's the head with these glycerol molecules and phosphate groups attached to them. And then those tails are hydrophobic, so they're going to want to be away from the water. And so what they'll do is they'll line up in a bilayer in two sets, and the heads will face the water and the tails will face in. And what this creates for us is what we refer to as the cell membrane. And that cell membrane is very good at regulating what moves from the outside of the cell to the inside of the cell and from the inside to the outside, because it's mostly hydrophobic, fat, water-fearing, fatty. And so most of our environment being wet and watery, it's going to automatically create a boundary between those two watery areas. The next group we're going to talk about is the steroids. These are significantly different from the phospholipids in that they're made of rings of carbon. They look something like this. They're all formed from cholesterol, which makes cholesterol of significant importance in our body. And examples of these molecules are things like testosterone and estrogen, so our sex-related hormones or gender-related hormones, and then also cortisol, which is our stress-related hormone. And these have really unique jobs in that they help our cells communicate with each other, create timing and regulation of activities in the body, and help our body respond to changes around us. You can see those 4 rings of carbon, 1, 2, 3, 4. And here each of the little green dots represents a carbon atom. Then there's other stuff hanging off of it, but specifically we're interested in those 4 rings of carbon because that's the signature for this particular molecule. Last group here that we're going to talk about is the fats and oils. And they're one that most people are more familiar with. We talk often about the difference between saturated fats and unsaturated fats and the health of those substances for us. And really to be a fat, a true fat, a biologically, chemically classified fat, you would have saturated fatty acids in you. And that's what makes you a fat. And to be a true oil, you're going to have unsaturated fatty acids. So this is a reference to that. Typically, the true fats are solid at room temperature and come from animal sources, though there are some exceptions. For example, the palm oils and coconut oil, which is a palm oil, are saturated fats that come from a plant. On the oils side, they're typically liquid at room temperature and come from plant sources. So all the examples you can come up with, olive oil and carrot oil and corn oil and canola oil and just vegetable oil, and walnut oil and peanut oil. These are all plant-sourced oils. And plants have those because they have unsaturated fatty acids in the structure of their fats. There are animal examples of unsaturated oils. Fish are a good example. They need unsaturated oils because they stay liquid at cooler temperatures, and a lot of fish live in much colder environments than a lot of land animals do. If you look at the structure of both fats and oils, you'll again see that glycerol backbone, and then 1, 2, or 3 fatty acids. And so if it has 1 fatty acid, we refer to it as a monoglyceride. If it has 2 fatty acids, we would refer to it as a diglyceride. And if it has 3 fatty acids, we would refer to it as a triglyceride. So that glyceride ending is in common with all of these fats and oils. And the more fatty acids that there are, the more energy there is for your body. But also, the more structure it has to break down. And you might go, well, more energy, that's definitely a good thing, right? Well, it can be a good thing, but it can also not be so great. And here's the reason why. When your body starts to break this apart, it's going to start pulling these hydrogens off of this saturated fatty acids. You see how there's hydrogens surrounding every single carbon here. That means this is saturated. So it starts ripping these off to harvest the energy in those bonds, right? And as it does that, now this hydrogen has to go somewhere. Well, where does it go? Well, it's that clingy girlfriend, remember, of chemistry, these lone hydrogens. So it wanders off and it attaches to the first thing it can. Well, first of all, if it wanders off and attaches to water, then basically what you're doing is you're forming acids. And then secondly, if it wanders off and attaches to something else, what it can sometimes form is what we call a free radical. And free radicals, we believe, are part of the aging system. They're molecules that have extra electrons on them. And so while they look like a normal molecule, they don't work in a chemical reaction the way they should, and they tend to gum up the works. They compete for spaces in chemical reactions where normal molecules should be. If, on the other hand, you go to break apart this unsaturated fatty acid in the lower part of this slide, you're not breaking hydrogens off the carbons. You're going to cut apart these carbons and harvest the energy that's there instead. And as you do that, you don't have to find a home for those hydrogens. They aren't there, and so they can't get lost and go off and form free radicals and acidify the body. So both things, both advantages of consuming unsaturated fats over saturated fats. And then there's one more representative of this family, the hydrogenated fatty acids. These are unsaturated fatty acids, like this one in the lower part of that slide, that have hydrogen added to them through chemical processes to become saturated. Margarine, Crisco, those kinds of fats are good examples of this. We do now make some margarines that do not use this process, and so they are actually truly oils stabilized by other compounds. But if what you're essentially doing is taking an unsaturated fat and turning it into a saturated fat, there's advantages there in that it becomes solid and more shelf stable and less temperature sensitive. But there's disadvantages in that you've changed the chemistry in a way that your body's then going to have to process more complexly. All right, that's it for our quick little overview of the carbohydrates and the lipids. In the next video, you'll see information about the proteins and the nucleic acids, and you're well on your way to getting ready for exam 1.